Hello guys, today our topic is irreversible cell injury. In previous lecture we have already discussed the reversible cell injury, causes of reversible cell injury and the mechanism of reversible cell injury. In today's lecture we will discussing about the irreversible cell injury. The first point about the irreversible cell injury is it lead to cell death. When the injurious stimuli if so severe, then the cell undergo irreversible cell injury. So, what are the causes of irreversible cell injury? Causes of irreversible cell injury. The number first cause is hypoxia. When hypoxia is so severe, cell will undergo irreversible cell injury. We also know that hypoxia lead to decreased ATP synthesis. When there is decreased ATP synthesis, cell will undergo irreversible cell injury. Two things are happen in irreversible cell injury. Number first is plasma membrane damage, and number second is mitochondrial dysfunction. Plasma membrane damage and mitochondrial dysfunction. Plasma membrane will be damaged in irreversible cell injury, and the second one is mitochondrial dysfunction. We will discussing these two separately. What occur in the mitochondrial dysfunction? Mitochondrial dysfunction. Mitochondria is so sensitive to many type of injurious stimuli like radiations, toxins, etc. When there is damage to mitochondria, it lead to the formation of permeability pores in the mitochondria. Pores are developed in the mitochondrial membrane. Permeability pores are developed in the mitochondrial membrane. So it will lead to, when pores are developed in the mitochondrial membrane, it will lead to the membrane potential dysfunction. Loss of membrane potential will occur. What means by loss of membrane potential? The cations and anions, sodium ions and potassium ion, calcium ion will enter into the mitochondrial membrane. Number second, we also know that mitochondria has cytochrome C. Mitochondria has cytochrome C. And we also know about the cytochrome C that in normal state it is the carrier protein in the electron transport change. But when mitochondrial membrane has become damaged, the cytochrome C is released into the cytoplasm. Released into cytoplasm. When cytochrome C is released into the cytoplasm, it causes internal cell injury. Cause internal cell injury. So two things are happened in the membrane dysfunction of the mitochondria. Number first, membrane potential is lost due to the permeability force produced in the mitochondrial membrane, and number second is cytochrome C is released into the cytoplasm, which cause the internal cell injury. Number second is membrane dysfunction. Membrane dysfunction. So when there is membrane dysfunction, membrane dysfunction, plasma membrane damage, it means plasma membrane damage occur. In irreversible cell injury, loss of membrane permeability occur. Loss of permeability will be occur. Plasma membrane permeability will be lost. When plasma membrane permeability is lost, entry of calcium ion into this plasma into the cell will occur. Entry of calcium ion, it means cytosolic calcium will be increased. When cytosolic calcium increase, it lead to activation of certain enzyme inside the cells. Increased calcium level led to activation of enzyme inside the cell. What are those enzymes? Number first. Number first is phospholipase. Due to increased calcium level, 5 phospholipase get activated. What is the function of phospholipase? Phospholipase break down phospholipids. It break down the phospholipids. Phospholipids breakdown occur due to the phospholipase enzyme. And when phospholipid breakdown occur, it lead to the formation of myelin figures. Myelin figures. It leads to the formation of myelin figures. So number first is formation of phospholipase occur due to the increased calcium level in the cytosol. Number second, endonuclease enzyme will be activated. Endonuclease enzyme will be activated due to the increased cytosolic calcium. What is the function of endonuclease enzyme? It damages the DNA. So phospholipid damage will occur breakdown. Number second, DNA damage will occur in irreversible cell injury. Number three, protease enzyme is activated due to intracellular cytosolic calcium increase. Protease enzyme will do protein breakdown. Protein damage will occur. Number four, RNase enzyme will be activated. RNase enzymes will be activated due to increased level of calcium in the cytosol. It will damage the RNA. So four things are happen. Number first, phospholipase will be activated, which will break down the phospholipids, and breakdown of phospholipid lead to the formation of myelin figures. Number second, endonuclease will be activated, which will lead to the damage of DNA. Number three, protease will be activated, which lead to the breakdown of lipids. And number four, RNase break down the RNA, damage the RNA. One extra clinical point you must be noted that in myocardial infection and a lot of disease, for example, myocardial infarction. In myocardial infarction, we measured troponin level. Why we measured this? We measured troponin level, yeah, or CKMB level. CKMB level. Why we made this in this disease? Because in this disease the cell injury occur. When the cell injury occur, plasma membrane dysfunction permeability for plasma membrane lose their permeabilities. When plasma membrane permeability is losses, this troponin and CKMB is get out from plasma membrane into the plasma. So we made this level, the level of troponin and CKMB. Similarly, in bile disease, we made the alkaline phosphatase level. This is the extra information about the mitochondrial membrane dysfunction. Number three things that happen in irreversible cell injury is lysosomal membrane damage. Lysosomal membrane damage. Membrane damage. When lysosomal membrane damage occur in the irreversible cell injury, activation lysosomal enzyme comes out into the cytoplasm. Lysosomal enzymes comes out into the cytoplasm. So because due to in the irreversible cell injury, the pH has been decreased due to the oxidative phosphorylation abnormality. When the pH is decreases, when the pH is decreases, it leads to the activation of activation of acid hydrolase. Acid hydrolase. Acid hydrolase. Acid hydrolase enzyme digests cell components. Digest cell components. We had already discussed in the irreversible cell injury that pH decrease occur due to the lactic acid accumulation inside the cell, which is occur due to the anaerobic glycolysis. So first thing happen that mitochondrial dysfunction will occur. Second is mem- plasma membrane damage occur, and third thing is lysosomal membrane damage. For MCQ's point of view, must note it that in mitochondria formation of flocculent amorphous densities occur. Amorphous densities appear. Densities appear. Number second, flocculent amorphous densities is the most important electron microscope morphological change in irreversible cell injury. Most important electron microscope morphological change in irreversible cell injury is the flocculent amorphous densities. Number third is most important light microscope features of irreversible cell injury. Feature of irreversible cell injury is nuclear change. We will be discuss the nuclear change inshallah soon. So what is nuclear change? What will be the change occur in the nucleus during irreversible cell injury? What change will occur in the nucleus? Number first, pyknosis. The first change in the nuclear change in the nucleus will occur pyknosis. Pyknosis is the clumping of nuclear chromatin. Nuclear chromatin will be clumped. Number second, karyorrhexis. Karyorrhexis. Karyorrhexis is the fragmentation of nuclear chromatin. Fragmentation of nuclear chromatin. And number third one is the karyolysis. 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 Karyolysis is the dissolution of nucleus. dissolution of nucleus. These three changes are occur in the nucleus during the irreversible cell injury. So we are going to the next concept which are given in the Robbin is oxidative stress. What is oxidative stress? Oxidative stress. It is just cellular abnormality, cellular abnormalities that are induced by reactive oxygen species. Reactive oxygen species. It means it just brings the concept of free radicals. Brings concept of free radicals. So, what is free radical? Free radicals. What are free radicals? Free radical is a molecule with one or more than one unpaired electron. Molecule with one or more than one unpaired electron. Because these are unpaired, they have unpaired electrons, so these are unstable. These molecules are unstable and produce lot of energy. Lot of energy. So what are free radical? Free radical are the molecule with one or more than one unpaired electron. Free radical mediated injury caused due to toxin, hypoxia, aging, and inflammation. These things lead to the free radical injury. Means in these cases free radical will be generated. Two concepts are given in the Robbin, that one is the generation of free radical. How free radical are produced? Generation of free radical. It means that how free radicals are produced. When there is radiation or toxin, when there is radiation or toxin, it lead to the formation of superoxide. It converts the oxygen molecule into the superoxide. Radiation or any type of toxin lead to conversion of oxygen molecule into the superoxide. Then enzyme comes called superoxide dismutase. It convert superoxide into the hydrogen peroxide. Superoxide into the hydrogen peroxide in the presence of iron. In the presence of iron, this is most important. And this reaction is called Fentose reaction. This reaction is called Fentose reaction. This reaction is called Fentose reaction. So, number first is radiation is generated the free radical. Number second, phagocytic leukocyte. Phagocytic leukocytes, for example neutrophils and macrophages. Normally ROS, reactive oxygen species, for example free radicals, these are generated in phagosomes. Phagosomes. These are generated in phagosome. So phagosome membrane contains certain enzyme that catalyze the superoxide and convert superoxide into the hydrogen peroxide. So how this occur in the phagocytic leukocytes. Superoxide is converted into the hydrogen peroxide. Then hydrogen peroxide in the presence of myeloperoxidase enzyme which is present in the leukocyte is converted into the hypochlorite. This is how the free radicals are generated, and we also know that in inflammation the leukocyte also release the free radicals. In inflammation the leukocyte also release the free radical. Number second concept is the removal of free radicals. Removal of free radicals. Cell also contains certain enzymes that remove the free radical, destroys the free radical to reduce the injurious effect. These are two types. Number first are enzymatic, and number second are non-enzymatic. By these two ways, cell remove the free radical or destroys the free radical. First, we will be discuss the enzymatic way. Enzymatic way. Number first is enzymatic way. In the cell there is presence of cell contain superoxide dismutase. What is the function of this enzyme? This enzyme destroys superoxide. This enzyme destroys superoxide. Remember, SUD is of two types. Number first is SUD1 and number second is SUD2. SUD1 is present in the cytoplasm of cell, and SUD2 is present in the mitochondria of the cell. What is the function of SUD? SUD destroys the superoxide free radical. For MCQs, remember that brain is protected from free radical injury, from free radical injury through SUD1, superoxide dismutase 1. This is the enzyme that protect the brain from the free radical injury. Number second is glutathione peroxidase. Glutathione peroxidase. This is found in cytoplasm of all cells. Cytoplasm of all cells. And what is the function of glutathione peroxidase? This enzyme destroys hydrogen peroxide. All these are important for the MCQ's point of view, that what are the function of glutathione, it inactivate the hydrogen peroxide, what is the function of sood, it inactivate the superoxide. And number 3, we all know about that, catalase present in peroxisome function, it convert hydrogen peroxide into water and oxygen. It means that it inactivate the hydrogen peroxides. What are the non-enzymatic way? Non-enzymatic way. In this, vitamin E, A, vitamin A, C, and beta carotene. This directly inactivate the free radical inside the cell. So, endoplasmic reticulum. Next concept is endoplasmic reticulum stress. What is endoplasmic reticulum stress? Endoplasmic reticulum stress brings concept of misfolding protein or unfolded protein. Unfolded protein. Endoplasmic reticulum has molecule called chapran. Chapran is basically control proper folding of protein during protein synthesis. What is the function of chapran? It control proper folding of protein during protein synthesis. If due to any toxin or radiation, protein are misfolded, misfolded. Just like cell, endoplasmic reticulum undergo adaptive response. Endoplasmic reticulum undergo adaptive response. First, endoplasmic reticulum will try to overcome this effect, which is called unfolded protein response. Which is called unfolded protein response, UPR. What will be happen in the unfolded adaptive response? In adaptive response, the endoplasmic reticulum start producing the chapran. Chapran production will be increased. Chapran production will be increased and translation will be decreased. So if translation will not occur, protein synthesis will not occur, will be not there. If protein synthesis will be not there, there will be less chance to produce misfolded protein. But if injurious stimuli is so severe, if injurious stimuli is so severe, it result in activation of pro-apoptotic sensors plus activation of caspases. Pro-apoptotic sensor will be discuss in the apoptosis. Now just believe me that it will lead to the cell death. It will lead to the cell death. So what is mean by this? The irreversible injury lead to the cell death, death of the cell. And cell death is of two types. One is the necrosis and other is the apoptosis. We will be discuss inshallah cell death in the next upcoming videos. Thank you.